Introduction. Definition of cell. It is basic unit of structure and function in living organisms. Explanation. 1. It is the smallest unit that can carry out all activities of life. 2. Cells are the building blocks of complex multicellular organism. Cell theory. 1. All living organisms are composed of one or many cells. 2. All new cells arise from by the division of pre-existing cells. 3. Cells contain the hereditary material of an organism which is passed from parent to daughter cells. 4. All metabolic processes take place within cells. Techniques used in cell biology. Microscopy. 1. Microscopy makes a very valuable contribution to our understanding of cells. 2. The techniques are also needed if the functions of organelles are to be studied. 3. Various techniques have been used for isolating and examining various cell components of these: differential staining, centrifugation, tissue culture, chromatography, and electrophoresis. 4. The most common techniques are: 5. The examination of a cell and its component depends upon the magnification powers of two convex lenses to produce a magnified image of a very small object. Resolution versus magnification of microscope. Magnification. Definition. Magnification means increasing the apparent size of the object. Explanation. MAGNIFICATION equals APPARENTSIZOFA and OBJCT over ACTULSIZ. 1. Magnification is the product of power of lenses used in a microscope, e.g. a compound microscope having 10x eyepiece magnifying power and 10x objective power will have 100x power. 2. With a light microscope, a specimen could quite easily be magnified by as much as 1,000 to 2,000 times. 3. The transmission electron microscope, TEM. The TEM can magnify an object up to 1 million times. Objective lens. Eyepiece lens. Magnification of object. X10. X6. X60. X10. 10x. 10x. 60x. X600. 10x. 100x. 1000x. 4. Scanning electron microscope, SEM, has produced three-dimensional images of whole objects. Resolution. Definition. The ability of microscope to distinguish details. Explanation. 1. The limitation of any microscope defined as resolving power. 2. The ability to distinguish close objects as being separate from one another. 3. The resolving power of light microscopes is 250 nanometers. 4. Its resolution power is about 500x more than human eye. 5. The human naked eye can differentiate between 2 points at least 1 millimeter apart. Staining. Definition. It is a process of treating a specimen for examination under the microscope with a reagent or dye. Explanation. 1. Biological structures are transparent, so to see them contrast is required. 2. Staining is used for contrast, to make cell organelles visible. Types of stains. Vital stains. Certain stains, when used in low concentration, are non-toxic to living tissues and can therefore be used on living materials. It can be applied to a living cell without killing them, e.g. trypan blue and Janus green B. Non-vital stains. A stain which is applied to dead cells is known as non-vital stain. These stains cannot be applied on living cells as these are toxic, e.g. eosin and aniline blue. Principal stains. The dye used to color the organelle of choice under study. Counter stains. A stain with color contrasting to the principal stain, making the stain structure easily visible using a microscope. Explanation. 1. It is the coloration of microorganisms by applying dye. 2. When a single dye is used, it is known as simple staining. 3. When two or more dyes are used, it is known as differential staining. For indifferential staining, the first basic dye is known as principal, primary stain, while another is known as counterstain. Profile of permanent stains. Strain. Final color. Suitable for aniline blue, blue, fungal hyphae and spores, borax carmine, pink, nuclei, obelia colony, eosin, pink/red, cytoplasm/cellulose, Fulgens stain, red-purple, DNA, particularly during cell division, i.e. chromosomes, Fleischmann's stain, red/pink/blue, blood cells, methylene blue, 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 nuclei, safranine, red/purple, nuclei, lignin and plant tissues. Profile of temporary stains. Stains. Final color. Suitable for aniline sulfate, yellow, lignin, iodine solution, blue-black, starch, Schultz's solution, chlorzinc iodate, yellow/blue/blue or violet, lignin, cutin, protein, /starch/cellulose. Different cell organelles stain differently by different stains, as shown in the table above. Increase a microscope's resolution power. Centrifugation, cell fractionation. Definition. Dividing the cell into its parts or fractions is called cell fractionation. Explanation. 1. It is done in a centrifuge and the process is called centrifugation. 2. Cell fractionation can isolate various components of cells including organelles according to their particular size and density. Process. This process is accomplished in the following manner. A. Formation of homogenate. The first step is grinding up the tissue in a liquid medium of proper osmotic concentration to form a homogenate. B. Centrifugation. 1. Homogenate is then put in a centrifuge and spun. 2. Initially it is spun at a relatively slow rate to separate out the larger, e.g. heavier part of the homogenate, such as any remaining whole cells and the nuclei. 3. The remaining material is again spun at a speed and cellular components of intermediate size, such as mitochondria, plastids etc. precipitate out and are removed. 4. The supernatant liquid can then be spun at still higher speeds and smaller, lighter cellular fractions are precipitated. Supernatant, the liquid lying above a solid residue after crystallization, precipitation, centrifugation, or other process. Tissue culture. Definition. Tissue culture is the growth of tissues and cells separate from the organism. Process. 1. This is typically facilitated by the use of liquid semi-solid growth medium such as broth or agar. 2. Tissue culture produces clones in which all the product cells are same. 3. The genotypes remain the same unless affected by mutation during culture. Genotype. A genotype is an individual's collection of genes. A tissue culture apparatus. Cell culture hood, i.e., laminar flow hood or biosafety cabinet. Incubator, humid CO, incubator recommended. Water bath. Centrifuge. Refrigerator and freezer, -20 degrees Celsius. Cell counter. Inverted microscope. Liquid nitrogen, GN, freezer or cryo storage container. Sterilizer, i.e., autoclave. B. Main steps in tissue culture. A. Selection of explant. 1. Selection of the plant tissue, explant, from a healthy vigorous mother plant. This is often the apical bud, but can be other tissue. B. Sterilization. 2. This tissue must be sterilized to remove microbial contaminants. C. Establishment. 3. Establishment of the explant in a culture medium. Medium sustains the plant cells and encourages division. It can be solid or liquid. For each plant species, and sometimes the variety within a species has particular medium requirements that must be established by trial and error. D. Multiplication. 5. Multiplication of the explant gives rise to a callus, a mass of loosely arranged and the auxin, low, cytokinin high ratios to form multiple shoots. 6. The callus may be subdivided a number of times. E. Application of plant hormones. 7. The shoots are transferred to a growth medium with relatively higher auxins, cytokinin ratios which results in the formation of roots. F. Deflasking, removal from the flask, and transfer to field. 8. Plants are deflasked and hardened off by gradually decreasing the humidity. This is necessary as many young tissue culture plants have no waxy cuticle to prevent water loss. Chromatography. Definition. Chromatography is a laboratory technique for the separation of a mixture. The mixture is dissolved in a fluid, gas or solvent, called the mobile phase, which carries it through a system, a column, a capillary tube, a plate, or a sheet, on which a material called the stationary phase is fixed. Explanation. 1. Chromatography is a mean of separating one type of molecule from others. 2. It involves moving the mixture, normally as a liquid or a gas, over a stationary phase embedded in cellulose or silica. 3. The separation may depend on a range of chemical and physical properties of the molecules such as solubility and molecular mass. Types. Essentially there are two basic ways of carrying out the separation. Paper chromatography. 1. It is used for the separation of photosynthetic pigments, sugar, or amino acids. 2. The mixture is spotted near one end of a paper strip and then dipped into a specific solvent which moves through the paper by capillary action carrying the molecules with it. 3. Similarly, a thin layer of silica may be used instead of paper. The difference between TLC and paper chromatography is that the stationary phase in TLC is a layer of adsorbent, usually silica gel, or aluminium oxide, and the stationary phase in paper chromatography is less absorbent paper. Column chromatography. It is more commonly used method which involves the mobile phase flowing over a supporting matrix held in a glass or metal tube. Electrophoresis. Definition. It is a technique used to separate molecules of different electrical charge under the influence of different electrical field. Explanation. 1. Negatively and positively charged ions, molecules, will move towards anode, plus, and cathode, respectively. 2. Two factors affect the speed with which charged molecules move towards an electrode. I. The amount of charge, greater charge, faster movement and vice versa. 2. The size of molecules, smaller molecules, faster movement and vice versa. Spectrophotometry. Definition. The technique that is used to measure the change in percentage transmission of light, optical density, of the suspension material. 1. Spectrophotometry is a tool that centers on the quantitative analysis of molecules depending on how much light is absorbed by colored compounds. 2. The cell mass is directly proportional to the optical density. 3. The determination of amino acids by spectrophotometric analysis is based upon specific light absorption. 4. The analysis of the amino acid generally depends upon absorption shown by their colored byproducts. Microdissection. Definition. The techniques in which microscope is used to help the process of dissection cells or its organelles. Explanation. Different kinds of techniques involve microdissection. Chromosome microdissection. 1. It is a technique that physically removes a large section of DNA from a complete chromosome. 2. The smallest portion of DNA that can be isolated using this method comprises 10 million base pairs, hundreds or thousands of individual genes. 3. A fine glass needle is used under a microscope to remove a portion from a complete chromosome. Laser microdissection. It is use of a laser through a microscope to dissect selected cells. Laser capture microdissection. 1. Laser capture microdissection is a contact and contamination-free method for isolating specific single cells or entire areas of tissue from a wide variety of tissue samples. 2. A laser is used through a microscope to cause selected cells to stick to a film. Micrometry, measurement of size with the microscope. Definition. Measurement of microscopic objects is called micrometry. Explanation. 1. In this method 2 scales are used. 2. One of the scales is placed in eyepiece, eyepiece gratitude, or ocular micrometer and other on the stage micrometer. 3. The micrometer has equally spaced divisions. 4. Eyepiece micrometer is first measured with stage micrometer and then eyepiece micrometer is used to measure microscopic structure. Cell wall and plasma membrane. Cell wall. Definition. A cell wall is a structural layer surrounding plant cells, just outside the cell membrane. Explanation. 1. The cell wall was discovered by Robert Hooke in 1665 earlier than the protoplast. 2. It is the outermost boundary of the plant cells. 3. It is secreted by the protoplasm of the cell, Golgi bodies. 4. Its thickness varies in different cells of the plant. 5. Each cell, whether isolated or occurring in tissues, has its own cell wall. 6. Cell walls of neighboring cells are held together by an intercellular substance called middle lamella. 7. Cell wall varies greatly in morphology and chemical composition, but its characteristics have a close relation with the age and function of the cell. Fundamental parts of cell wall. The cell walls have 3 fundamental parts, namely: I. Middle lamella. 2. Primary wall. 3. Secondary wall. I. Middle lamella. 1. Middle lamella cements together the walls of the neighboring cells. 2. It is usually thin and about 1 m in thickness. 3. In woody tissues the middle lamella is commonly lignified. Contain lignin. 2. Primary wall. 1. Primary wall is the first wall formed in a developing cell. 2. It is usually 1 to 3 m in thickness. 3. It is elastic and extendable, crystalline, and optically active. For optically active compound is capable of rotating the plane of vibration of polarized light to the right or left. 5. It is composed of cellulose, pectic compounds, from Greek pectikos coagulating. 6. Pectin is a structural acidic heteropolysaccharide contained in the primary and middle lamella and cell walls of terrestrial plants. Its main component is the sugar acid derived from galactose. 7. It also contains polysaccharides and hemicellulose. 3. Secondary wall. 1. The secondary wall follows the order of primary wall in development. 2. It is laid down inside the primary wall. 3. Its thickness is about 5 to 10 m, more or less rigid, crystalline, and strongly optically active. 4. It is mainly composed of lignin, cellulose, non-cellulosic polysaccharides, hemicelluloses, and mineral salts of Ca, Mg, K, and some silica. 5. Lignin, a complex organic polymer deposited in the cell walls of many plants, making them rigid and woody. 6. A major role of the cell wall is to protect cells from overexpansion. 7. Plant cell wall maintain the shape of cell. Plasma membrane. Definition. The plasma membrane, also called the cell membrane, is the membrane found in all cells that separates the interior of the cell from the outside environment. Explanation. 1. The plasma membrane consists of a lipid bilayer that is semi-permeable. Differentially permeable allows nonpolar but stop-polar compounds. 2. Semi-permeable allowing certain substances to pass through it but not others, especially allowing the passage of a solvent but not of certain solutes. 3. The plasma membrane controls the transport of materials entering and leaving the cell. 4. Cell membrane is chemically composed of lipids, 20 to 40%, and proteins, 60 to 80%. 5. In addition there is small quantity of carbohydrates present. 6. In 1972, Singer and Nicholson proposed a most acceptable model for membranes called fluid mosaic model. 7. This model is in agreement with photograph of cell membrane by electron microscope. 8. This model explains that the membrane is like a sea of lipids in which protein are floating. A role of proteins. 1. The globes of proteins are moving about in the sea of lipid molecules. 2. Intrinsic proteins penetrate and bind tightly to the hydrophobic tails of lipid bilayer. 3. Extrinsic proteins are loosely bound to the hydrophilic, polar, glycerol head surfaces, which face the watery medium both inside and outside the cell. 4. Intrinsic proteins extend completely through the double layer of lipids from one side to the other. 5. Extrinsic proteins are on one side of the membrane. 6. Carbohydrates extend out from the outer surface of the membrane and are attached either to membrane lipids as glycolipids or to proteins as glycoproteins. B. Role of glycolipids and glycoprotein. 1. Lipids, proteins and carbohydrates are responsible for functional diversity of the membranes. 2. Lipid bilayer makes the membrane differentially permeable barrier that allows the transport of nonpolar materials across it and prevents ionic materials. 3. Membrane proteins on the other hand makes it selectively permeable barrier that select materials according to cell's need. 4. Extrinsic proteins function as receptor that receives the stimuli from the environment and thus inform the cell to respond. 5. Integral proteins also called as permeases regulate diffusion, osmosis and active transport of ionic materials. 6. Membrane carbohydrates such as glycolipids and glycoproteins provide receptor sites that receives different types of stimuli like hormone receptor sites, HRS, receives nerve impulses, recognition of antigen in food materials thus inform the cell for response. 7. Membrane carbohydrates are also responsible for endocytosis i.e. phagocytosis, eating of the cell, and pinocytosis, drinking of the cell. 8. Glycolipids and glycoproteins act as cell surface markers. 9. Cell surface markers. Cell surface markers are special proteins expressed on the surface of cells or carbohydrates attached to the cell membrane that often conveniently serve as signs of specific cell types. Role of plasma membrane. 1. Plasma membrane is a dynamic structure only about 7 nanometers wide. 2. It present barrier to the movement of ions and molecules. 3. It prevents the aqueous contents of the cell from escaping. 4. It helps to excrete waste substances, urea, uric acid etc. 5. It also secrete useful substances, hormones, enzymes, etc. 6. Cell membrane generate the ionic gradients essential for nervous and muscular activity. 7. It maintain a suitable pH and ionic concentration within the cell for enzyme activity. Cytoplasm. Definition. Cytoplasm is a thick solution that fills each cell and is enclosed by the cell membrane. Explanation. 1. The cytoplasm substance contain a variety of parts. 1. Cell organelles. 2. Insoluble wastes. 3. Storage products. 2. Cytosol. The cytosol is the aqueous portion of the cytoplasm in an intact cell. 1. It consists of water, organic molecules and dissolved ions. 2. The soluble part of the cytoplasm forms the ground substance between the cell organelles and is called cytosol. 3. It is formed of about 90% water and forms a solution which contains all the fundamental biochemical of life. 4. In cytosol, small molecules and ions may form true solutions, and some large molecules form colloidal solutions. 5. Colloidal solutions, colloids, also known as colloidal solutions or colloidal systems, are mixtures in which microscopically dispersed insoluble particles of one substance are suspended in another substance. 6. A colloidal solution may be a sol, non-viscous, or a gel, jelly-like or viscous. Often the outer region of cytoplasm is more gel-like. 7. Sol colloidal solution. A sol is a colloid made out of solid particles in a continuous liquid medium. Sols are quite stable. Examples include blood, pigmented ink, cell fluids, paint plant cytoplasm. 8. Gel colloidal solution. A solid or semi-solid state of colloidal solution, e.g. animal cytoplasm or ectoplasm. 9. Storage products and metabolic pathways. Apart from acting as storage of vital chemicals, the ground substance, cytosol, is the site of certain metabolic pathways, such as glycolysis. 3. Cell organelles. In living cells, the cytoplasm contains several cell organelles such as endoplasmic reticulum, mitochondria, Golgi complex, nucleus, plastids, ribosomes, lysosomes, centrioles, and various others. 4. Cytoplasmic streaming or cyclosis. The flow of the cytoplasm inside the cell, driven by forces from the cytoskeleton. Endoplasmic reticulum. Definition. A network of membranous tubules within the cytoplasm of a eukaryotic cell, continuous with the nuclear membrane. It usually has ribosomes attached and is involved in protein and lipid synthesis. Explanation. 1. It is a network of channels run through the cytoplasm of all the eukaryotic cells. 2. These channels are often continuous with plasma membrane and also appear to be in contact with the nuclear membrane. 3. The whole system of channels is said to be endoplasmic reticulum. 4. These membranes vary widely in appearance from cell to cell. 5. These channels are separated from the cytoplasmic materials by the spherical or tubular membranes one above the other, called cisternae, singular, cisterna. 6. Cisternae. A cisterna, plural, cisternae, is a series of flattened, curved membrane saccules of the endoplasmic reticulum and Golgi apparatus. Types of endoplasmic reticulum. There are two morphological forms of endoplasmic reticulum. 7. Rough endoplasmic reticulum. 1. It is with attached ribosome. 2. Rough endoplasmic reticulum, RER, are mostly present outside the nuclear membrane and thus involved in protein synthesis. 3. After synthesis, the proteins are either stored in the cytoplasm or exported out of the cell through ER. 8. Smooth endoplasmic reticulum. 1. These are without ribosomes. 2. Lipid synthesis. These help in metabolism of a number of different types of molecules, particularly lipid synthesis. 3. Detoxification. They also help to detoxify the harmful drugs. 4. Transmission of impulses. In some cells, SAR is responsible for the transmission of impulses, for example, muscle cells, nerve cells, etc. 5. Cell-to-cell transport. SAR play an important role in the transport of materials from one part of the cell to the other. 6. Desmotobule is an endomembrane-derived structure of the plasmodesmata that connects the endoplasmic reticulum of two adjacent plant cells. 7. It provides mechanical support to the cell so that its shape is maintained. Ribosomes. Definition. A minute particle consisting of RNA and associated proteins found in large numbers in the cytoplasm of living cells. They bind messenger RNA and transfer RNA to synthesize polypeptides and proteins. Explanation. Size. These are tiny cell organelles, about 20 nanometers in diameter. Discovery. These were first studied by Pallotti, 1955. Structure. Eukaryotic ribosomes are composed of almost equal amount of RNA, ribonucleic acid, and protein, hence they are ribonucleoprotein particles. The RNA present in ribosomes is ribosomal RNA. Ribosomes exist in two forms, either freely dispersed in the cytosol or attached with RER as tiny granules and are the site of protein synthesis. Ribosomes are synthesized in nucleolus of the nucleus. An example of protein synthesized by free ribosomes is hemoglobin in young RBCs. Each ribosome consists of two subunits, one large and one small. Sedimentation has revealed two basic types of ribosomes called 70S, 50SAN30S, and 80S, 60S and 40S, ribosomes, S equals Svedberg unit used in ultracentrifugation. The 70S ribosomes are found in prokaryotes while slightly larger 80S in eukaryotes. The two subunits on attachment with each other require Mg ions. The ribosomes are attached to mRNA through small ribosomal units. A group of ribosomes attached to mRNA are called polysomes. The Golgi complexes. Definition. It consists of stacks of flattened, membrane-bounded sacs called cisternae. These cisternae together with associated vesicles are called Golgi complex. It is a complex system of interconnected tubules around the central stack. Explanation. Discovery. The Golgi apparatus was discovered by Camillo Golgi in 1898, using special staining techniques. Occurrence. This apparatus, which was found virtually in all eukaryotes. Golgi complex in plants. In plants it consists of units called dictyosomes. Each dictyosome is formed of bundles of bent and flattened cisternae-associated tubules and secretory vesicles. Faces of dictyosomes. Dictyosome has two distinct faces. The proximal, cis, or forming face present close to nucleus. Distal, trans, or maturing face located towards the cell membrane. Formation of new cisterna, vesicles and tubules pinched off from RER to flow, converge and fuse with the forming face to form new cisterna. Functions of Golgi apparatus. Storage of secretory products. Golgi apparatus helps in the storage of secretory products. Modification and packaging. It helps in the modification and packaging of secretory products. Synthesis of polysaccharides. In some cases, polysaccharides may be synthesized from simple sugars, disaccharides from SR, in the Golgi apparatus. Formation of glycolipids and glycoproteins. These polysaccharides may then be attached to proteins and lipids to form glycoproteins and glycolipids. Addition to the plasma membrane surface, secretory vesicles produced by the Golgi apparatus may play an important role in adding surface area to the plasma membrane. Lysosomes. Definition. They are surrounded by a single membrane and are simple sacs that contain very large variety of food-digesting enzymes called hydrolases. Explanation. Meaning of lysosomes. Lysosomes, lyso-splitting, soma body, are cytoplasmic organelles. Occurrence, found in cytoplasm of most eukaryotes. These are different from other cytoplasmic organelles due to their morphology. Discovery. These were isolated as separate components for the first time by de Duva in 1949. Cell digestion. Any foreign objects that gain entry within the cell are immediately engulfed by the lysosomes and are completely broken down into digestible pieces. Phagocytosis. This process is known as phagocytosis. Abundance. They are very abundant in those animal cells which exhibit phagocytic activity, example neutrophils. Contents. Lysosomes are bounded by single membrane and contain numerous hydrolytic and acid phosphatase enzymes. Acid phosphatase, AP, is an enzyme that catalyzes the hydrolysis of phosphate esters in an acidic environment. Hydrolytic enzymes. Lysosomes are membrane-enclosed compartments filled with hydrolytic enzymes that are used for the controlled intracellular digestion of macromolecules. They contain about 40 types of hydrolytic enzymes, including proteases, nucleases, glycosidases, lipases, phospholipases, phosphatases, and sulfatases. Synthesis. These enzymes are synthesized on RER and are further processed in the Golgi apparatus, MCQ. Primary lysosomes. The processed enzymes are butted off as Golgi vesicles and are called primary lysosomes. These contain those enzymes which can digest the phagocytosed food particles. Secondary lysosome. A lysosome formed by the combination of a primary lysosome and a phagosome or pinosome and in which lysis takes place through the activity of hydrolytic enzymes. Residual bodies. Residual or tertiary lysosomes. They are those lysosomes in which only indigestible food materials have been left. The residual bodies or lysosomes pass outwardly and fuse with the plasma membrane to throw out the debris into external environment by exocytosis or effaggy. Autophagy. During autophagy, self-eating, some old worn-out parts of the cell are also digested. Recyclers of the cells. Materials of cell may be recycled, and cell may be renewed. Suicide bags and developmental process. Their enzymes can also result in degeneration of cell, as may occur during the developmental processes. Malfunctioning of lysosomes. Congenital, inborn, diseases. Several inborn diseases have been found to be due to accumulation of substances such as glycogen or various lipids within the cell. Storage diseases. These are produced by a mutation that affects one of the lysosomal enzymes involved in the catabolism of certain substances, for example. Glycogenosis type 2 disease. Tay-Sachs disease. Glycogenosis type 2 disease. Overstorage of glycogen. The liver and muscle cells appear filled with glycogen within membrane-bound organelles. Enzyme responsible. In this disease, an enzyme that converts glycogen to glucose is absent. Background of glycogen storage disease. Glycogen storage disease type 2, GSD2, also known as Pompe disease, is part of a group of metabolic diseases called lysosomal storage disorders, LSDs. GSD2 is an autosomal recessive disorder that results from deficiency of acid alpha-glucosidase, also known as acid maltase, a lysosomal hydrolase. The cellular role of acid alpha-glucosidase is to convert glycogen into glucose within the lysosomes. Tay-Sachs disease is involved in the catabolism of lipids. Accumulation of lipids in brain cells lead to mental retardation and even death. Overview: Tay-Sachs disease is a rare genetic disorder passed from parents to child. It's caused by the absence of an enzyme hexosaminidase, hexa, that helps break down fatty substances. These fatty substances, called gangliosides, build up to toxic levels in the brain and spinal cord and affect the function of the nerve cells. Peroxisomes. Definition: the peroxisomes are small subcellular bodies approximately 0.5 in diameter, surrounded by membranes. Explanation: occurrence: these are found in a great variety of organisms, including plants and animals, which are also called microbodies. In animals, they are most common in liver and kidney cells. Denser than lysosomes, they are similar to lysosomes, but they are somewhat denser and have different enzyme systems. Enzymes of peroxisomes. The enzymes of peroxisomes are known to break down hydrogen peroxide into oxygen and water, protecting cells from its destructive effects. Liver peroxisomes contain 4 enzymes related to the metabolism of H2O2. 3 of them are urate oxidase, diamino oxidase, and hydroxylic acid oxidase, which produce peroxide, H2O2, and 4th is catalase, which destroys peroxide. Catalase is found in the matrix of peroxisomes and represents up to 40% of the total protein. H2O2 is toxic, so catalase destroys it, and it probably plays a protective role. In the leaves of green plants, it is involved in photorespiration. Glyoxysomes. Definition: the glyoxysome is a plant peroxisome, especially found in germinating seeds, involved in the breakdown and conversion of fatty acids to acetyl-CoA for the glyoxylate bypass. Since it is also rich in catalase, the glyoxysome may be related to the microbodies or peroxisomes or derived from them. Explanation: glyoxysomes are a type of peroxisomes that are found in plants, especially in the fat storage tissues of germinating seeds. Seeds that contain fats and oils include corn, soybean, sunflower, peanut, pumpkin and castor etc. contains glyoxysomes. Just like in peroxisomes, in glyoxysomes the fatty acids are oxidized to acetyl-CoA by peroxisomal beta-oxidation enzymes. This process helps in seed germination. These organelles are absent in seeds poor in lipid. Cytoskeleton. Definition: the cytoskeleton is a network of filaments and tubules that extends throughout a cell, through the cytoplasm. Explanation: Koltsov in 1928, by his microscopic studies, suggested the existence of an organized fibrous network of skeleton in the cytoplasm of eukaryotic cells. Cohen, 1977, confirmed the views of Koltsov by his electron microscopic studies. It is cytoplasm of eukaryotic cells contains a cytoskeletal network of different types of microtubules, microfilaments and intermediate filaments. Cytoskeleton consists of 3 types of elements: microtubules, microfilaments and intermediate filaments. They are visible only with the help of electron microscope. They are absent in prokaryotic cells. Microtubules and microfilaments are dynamic structures, constantly forming and disassembling. Location: they are found in the cytoplasmic matrix of all eukaryotic cells. They are also present in structures like centrioles, basal bodies, cilia or flagella, sensory hair, spindle apparatus, chromosome fibers, nerve processes, sperm tail etc. Structure and composition: the main proteins that are present in the cytoskeleton are tubulin, in microtubules, actin, myosin, tropomyosin and others which are also found in muscles. Functions: cyclosis and amoeboid movements are because of microfilaments. Intermediate filaments are involved in determination of cell shape and combination of cellular components. Types: A. Microtubules. Microtubules contain some longitudinally arranged assemblies of filaments. These filaments are known as protofilaments. About 13 protofilaments form a hollow cylinder that are recognized as microtubules. These are long, unbranched slender tubulin protein structures. Function of microtubules. One very important function of microtubules is their role in the assembly and disassembly of the spindle structures during cell division. B. Microfilaments. These are considerably more thin cylinders made up of contractile proteins, linked to the interface of the plasma membrane. They are involved in cyclosis. C. Intermediate filaments. This type of cytoskeletal element is made of multiple strands of fibrous proteins wound together. As their name suggests, intermediate filaments have an average diameter of 8 to 10 nanometers, in between that of microfilaments and microtubule. They help in maintaining the cell shape.